Good morning to everyone from US and good evening from everybody in India. Today we announced the beginning of a seminar series, the result of a dynamic collaboration between the School of System Science and Industrial Engineering at Binghamton University and the School of Healthcare Science and Engineering Initiative at Vellore Institute of Technology. We plan to have this partnership to bring together leading minds, innovative research, and diverse perspective from two renowned institutes dedicated to advancing knowledge and transforming the future of systems engineering and interdisciplinary innovation. We welcome students, researchers, and professionals from across discipline to participate in this enriching series as we continue to build bridges between academic excellence and real-world impact. I would like to invite Dr. Kasavani to have a few words to felicitate this event.
Good morning, everyone, and good evening to our colleagues in India. It is a great pleasure for me to be here with all of you today. My name is Mohammad Kasavani. I am the director of the School of System Science and Industrial Engineering at Binghamton University. And on behalf of Binghamton University, the State University of New York, I would like to welcome all the participants from VIT and from Binghamton University to today's inaugural seminar. We're delighted to have this partnership with VIT. And as some of you may already know, the partnership between Binghamton University and VIT Vellore in particular started almost 15 years ago. And over the last decade and a half or so, I have personally had the pleasure to visit at least 3 of the VIT campuses. And we've had the great pleasure to host Chancellor Viswanathan on our campus, I think 3 or 4 times. And he has also received an honorary PhD from Binghamton University. And we are delighted that as part of this partnership, we are able to collaborate with great faculty and host wonderful students and scholars from VIT. And we are delighted to continue to expand our partnerships within the VIT group of institutions. And one of the things that has a special place in our partnership is the School of Healthcare Science and Engineering. Personally, for me, because I serve as the Director of Healthcare Systems Engineering, there is significant synergies between our research center and the expertise of our faculty and SHINE. And we are delighted that as part of this partnership, we are able to offer a joint seminar series to kick off this partnership. We have plans for many activities, including faculty exchanges, student exchanges, and joint seminars, in addition to other activities that you will get to hear about in the near future. And that's why I'm delighted that we are kicking off this event by a seminar by one of our wonderful colleagues in the School of System Science and Industrial Engineering. And before we go ahead and turn it over to our colleagues at VIT, I would like to thank Dr. Geeta for her partnership and collaboration. And I would like to thank Dr. Yogendra as well and all the other faculty who are here today for coordinating this event and for participating. And thank you again for the partnership and look forward to all the collaborations. Thank you.
Thank you, Dr. Kasavani. Now I would like to invite Dr. Yogendra Pratap Singh to have a few words before we start the seminar series.
Thank you, Dr. Srinath, and thank you, Professor Kasavani, for this wonderful, kind words. At the very outset, I'd like to highlight one point. What we discussed was if this seminar series would broadly be coming under Shine X Talk series. So we are starting this talks seminar series, and then this was, interestingly, the inaugural session of that. So firstly, let me make that announcement that being the inaugural session, I welcome you all once again, both from Binghamton side and VIT side. At the outset, let me extend my sincere thanks to Professor Geeta Manivasagan, who's the Dean of Shine at VIT Vellore here. And she is just finishing a management meeting. She'll just be joining us, but she's given her wishes. And then let me thank Dr. Krishnaswamy Srihari, who's been very instrumental in all these collaborations. Of course, Professor Kasavne and Dr. Srinath has been there in really taking this forward. So, as Professor Kasavne put it, this is just the beginning. We have much activities to do together. With that, I think we can start the seminar series. Thank you once again.
Yes, thank you, Dr. Yogendra. With that, I would like to invite Dr. Narmada Sambaturu, who is our first speaker for the seminar series. So she's an assistant professor in the School of Systems Science and Industrial Engineering at Binghamton University. Prior to this, she was a director's postdoctoral fellow at Los Alamos National Lab and obtained her PhD in mathematical biology from Indian Institute of Science. She holds an MSc in computer science from the National University of Singapore and Bachelor of Engineering in Computer Science and Engineering from Visvesvaraya Technological University in India. Her research involves developing novel, multi-scale and data-driven methods to study infectious diseases in the context of heterogeneous host immune responses. Her work cuts across multiple disciplines and leverages a wide array of tools, including mathematical modeling, machine learning, genomics, system biology, and epidemiology to answer questions in public health. Without further ado, I invite Dr. Narmada. Dr. Narmada, the floor is all yours.
Thank you, Dr. Srinath, and thank you, Dr. Kasavne and Dr. Yogendra, for organizing this series. And I'm really honored to be the first person to be speaking in this series. I hope I do it justice. I'll share my screen now. Please let me know if you have any problems seeing the screen.
Yes, we can see the screen.
Excellent. All right, so let's get started. Before I begin, I'd like to briefly mention my journey to multi-scale modeling and combining healthcare and engineering together. As Dr. Srinath mentioned, I started with a bachelor's of engineering in computer science and engineering in Ramaya in Bangalore. And while I was doing this, I wasn't really sure what I wanted to do in life. All I knew was that I wanted to do research. And I started doing a few internships here and there. And one of those internships involved bioinformatics. By the end of my bachelor's, I still wasn't sure what I wanted to do. So I went with the crowd. I joined Tata Consultancy Services as a developer. But while I was in TCS, one of the work I had done during my bachelor's, the bioinformatics project, that got published. And I started reading more about bioinformatics and biology. And that got me excited, which made me join a master's in the School of Computing in National University of Singapore, where my focus was on genomics. I figured, okay, genomics is the buzzword these days. Let me experience what it's like to study genomics and apply techniques from computer science to study that. I then moved on to a PhD in Indian Institute of Science to do mathematical biology, where I thought, why just engineering? Maybe we can apply math as well and answer questions in biology. So it's been a little bit of a meandering road. But during my PhD, I was able to visit the University of Leeds, the Department of Applied Math. And bringing all of this together, the expertise from computer science, mathematics, I was able to win a director's postdoctoral fellowship in Los Alamos, where I started working on HIV. So through this journey of engineering and math and applying all of these to answering questions in biology and in healthcare, I have been very fortunate to have been accepted as an assistant professor at Binghamton University at the School of Systems Science and Industrial Engineering. And I've had a great time working here, and I hope that all of you can see the value of this collaboration as well. Without further ado, let's start. So infectious diseases is one of the main things I'm interested in. And how do infectious diseases spread? They spread when you have an infected individual and a bunch of susceptible individuals. And the infected individual, say, sneezes, and spreads particles of the pathogen, depicted here by these red devil-like creatures, into the atmosphere, and the susceptible people come in contact with it. So you can see that there is an assumption of homogeneity in this scenario. That is, we're assuming that all the susceptible individuals are the same as each other. But the truth is that heterogeneity exists. In this cartoon, this person with no armor is more susceptible. This person with a very strong armor is very immune, not at all susceptible. And when such heterogeneity exists, which of these individuals catches the disease changes, and how the whole population responds to a disease, that changes too. So at a population scale, although pathogen exposure may be the same, the heterogeneity in the susceptibility can affect how many people actually get infected, how large an epidemic will be. But to understand this, we need to understand the source of this heterogeneity. And if we investigate the source of the heterogeneity, we need to start looking at each individual separately. Within each individual, that is, within the host, there is an interaction between the immune system and the pathogen that they're exposed to. And if the immune response is strong, the person will not get the disease. If the immune response is weak, the person may get the disease. So the strength of the immune response is what determines how susceptible a person is to getting a disease. But how do we understand how strong the immune response is? That depends on the interaction between the immune cell and the virus or the infected cell. Specifically, the molecular scale interaction between the molecules of the immune system and the molecules of the pathogen. So you can see that there are so many different scales that are involved in determining how an epidemic spreads, how an epidemic takes place. There's the interaction at the molecular scale, the within host systemic interaction, and a population scale spread of the disease. So to figure out what is happening, we need to treat an infectious disease as a complex system, which it is, where a set of interacting parts result in an emergent behavior, which is more than the sum of those interactions. So treating infectious diseases as a complex system and embracing their multi-scale nature, we can start to understand not just what is going to happen, but why it happens. So using multi-scale modeling, we can obtain novel insights, which are explainable and actionable. And I hope I can demonstrate to you that that's the case. And for me, the most important thing is that it's a lot of fun. I'd like to explain two projects to you. The first one is host and pathogen genetic heterogeneity in epidemic models. And it has been identified as a key challenge in epidemic modeling to incorporate genetic information to figure out what happens at an epidemic scale. So we have this system where we have interactions at the molecular scale, within host scale, and population scale, determining how a population responds to an epidemic. In order to model this, the thought process I use is examine the interface between the scales. So in this case, let's start with the interface, which is between the immune system and the pathogen, which occurs at the molecular scale. To dive a bit deeper, the way the immune response to a viral infection like influenza or SARS-CoV-2, the way the immune response works is a host cell gets infected. The virus actually enters into a host cell and uses the host cell's mechanisms to do its work. So within that infected cell, we'll have peptides, that is protein fragments from the virus. An immune cell, particularly a CD8 plus T cell, is an important part of the response to such an intracellular virus. The immune cell has toxic enzymes within it, which it can release to combat the virus. The immune cell and the infected cell interact through this particular junction, which is between an HLA class 1 molecule and a viral peptide and a part from the T cell as well. Let's focus in on this HLA class 1 molecule because this is an important bottleneck in this presentation. What happens is every cell in our body expresses HLA class 1. CD8 plus T cells, that is cells from the immune system, roam around in the body, patrolling every cell that they encounter, which expresses HLA class 1. So the immune cell actually rolls around every single cell which has HLA class 1 and samples the peptides presented on them. If that peptide is recognized as non-self, that is, it's a virus or any other thing that shouldn't be there, the immune cell gets activated. Once it's activated, the immune cell breaks away from the infected cell and releases toxic enzymes, resulting in the death of the infected cell. So the conditions for a successful CD8 plus T cell response are that the peptide or the protein fragment from the virus must be bound to an HLA class 1 molecule, and the presented peptide must be recognized as non-self. So only if both of these conditions can a strong immune response be mounted, if these conditions are satisfied. So we made a hypothesis that the susceptibility of an individual is inversely proportional to the number of epitopes presented by that individual. Epitope is just another name for this peptide or protein fragment that is bound to HLA class 1. So I'll be using the term epitope going forward. So the logic behind this is if a large number of epitopes from the virus can be presented to the immune system, it's very likely that the immune system will recognize at least some of them as non-self and mount an immune response. So the person is less susceptible. However, if almost no epitopes from the virus get presented to the immune response, the immune response has no chance of recognizing it, let alone killing the virus. So the susceptibility of an individual is inversely proportional to the number of epitopes from the virus that can be bound to HLA class 1. It turns out in each of us, we have not just one HLA class 1 molecule, but rather an HLA genotype. That is, a person has 6 HLA class 1 alleles. Allele just means variant. So there are 6 variants, 2 each of type A, B, and C. Now it turns out that there are databases out there. An example is the Allele Frequency Net database, in which people have gone out into the world, sampled blood from different people, and actually measured what HLA class 1 alleles they have. We then get a proportion. Say 20% of this population has this particular variant. 1% of this population has this particular variant, and so on and so forth. So we can essentially draw something like a pie chart among HLA class 1A variants in, say, Bangalore or in Vellore. How many people, what percentage of the people have this particular variant? What percentage of the people have this particular variant, and so on. So you get a pie chart of percentages for HLA-A, percentages for B, and percentages for C. Having acquired such percentages, we can now generate HLA genotypes. That is, we can sample 2 A alleles, 2 B alleles, and 2 C alleles in a manner proportional to their frequencies. When we do this, we can generate a synthetic population, a list of people essentially represented by their HLA genotypes. Having done that, we can now predict for each of these people how many epitopes can be presented by their HLA. And the total number of epitopes is the total number of epitopes presented by each of the alleles in that genotype. So remember I said we can predict how many epitopes can be presented. So that is exactly what we're doing. We're predicting, given a particular variant of HLA and a particular protein from the pathogen. So if it's SARS-CoV-2, we take the surface protein and we say, okay, given the population in Vellore, this particular allele, HLA allele, is common. So people who have this variant, how many proteins from SARS-CoV-2 can this person present? To do this prediction, we use machine learning. We use a combination of neural networks, stabilizing matrix, and combinatorial peptide library, and only take those results which are predicted to have high binding affinity by all three methods. So that gives us a predicted list of epitopes that each person can present. So using this molecular scale interaction, we have now been able to estimate the susceptibility of each person. How susceptible is each individual in a given population? But the next step to predict how an epidemic would spread, we need to also think about how individuals interact. So going back to our mantra, the thought process is to examine the interface between the scales. How do people interact with each other? In classical epidemic models, such as the SIR or susceptible, infected, recovered models, the assumption is that all people are equally likely to interact. So every pair of people is as likely to interact as any other pair. This is a well-mixed population. Having made that assumption, people are classified according to their state with respect to the disease. They are classified as being susceptible, infected, or recovered. Then we model the transition from susceptible state to infected state and transition from infected state to recovered state, almost like flowing through a pipeline. If the rate of transition from S to I is large, it's like many people flow from the S state to the I state and so on. In this project, we chose to keep the assumption of well mixed, but we know that we have the estimated susceptibility of each person based on their genetics. So we can now group people based on how susceptible they are. So we get a bunch of susceptible subgroups. And for each susceptible subgroup, the rate of transitioning to the infected state will be different. For this group with no armor, who are very susceptible, the rate of transitioning to infected will be high. For this group, which is less susceptible, the rate of transitioning to infected will be low. So we now have everything we need. We did our modeling using the inputs of H1N1, which is a type of influenza A. This was actually the swine flu, which caused a pandemic in 2009. So we have 81 strains of this swine flu isolated in 2009 and 85 strains isolated in other years. We also look at HLA allele frequencies for 61 ethnicities from the database I mentioned earlier. The predicted values are the susceptibility of each individual, of course, and then how the final epidemic size will be. And we found that by and large, the predictions match the observed incidence during the 2009 swine flu pandemic. Now, you might wonder, why focus on influenza? Why 2009 specifically? So this is the CDC website for the US, in which they track the flu seasons and which particular strains are common in each season. This is for January 2025, so earlier this year. And it was found that influenza A, specifically the pandemic 2009 strain, was one of the predominant viruses reported in that week. And this was actually one of the predominant viruses reported in this year's flu season overall. So influenza A, the 2009 strain, is actually a very important strain even today. One of the insights that we got from doing this prediction was as follows. In this plot, on the x-axis, you see the average susceptibility of the population. So suppose Vellore, maybe the average susceptibility is very low. So the population would lie on the left side of the x-axis. On the y-axis, you see the coefficient of variation. That tells you how spread out are the susceptibility values within that population. So the average may be 0.5, but maybe in Vellore, everybody has an average close to 0.5, but maybe in Chennai, people have a range of values. Some people have value 2, some people have value 1, some people have value 0.25, etc. So the coefficient of variation tells you the spread of susceptibility values. The color in each circle is the epidemic size that is predicted. So a more red color indicates a larger epidemic, and a more blue color indicates a smaller epidemic. So let's focus in on this region where the average susceptibility is medium, a mid-range of average susceptibility. As you go up on the y-axis here, you see that the color of these circles changes from being very red to being more blue. That is, as the coefficient of variation increases or the spread in susceptibility values increases, the predicted epidemic size decreases. That is, diversity protects a population. So a population which is well mixed, not just in terms of movement of people, but in terms of the genetic composition, can actually protect the whole population and make it such that the population experiences smaller epidemics, even though on average, the susceptibility may be somewhere in the middle. So we thought that was a pretty cool prediction. But because we did our modeling with biological insights, we can go deeper than this. We can explore why. For example, in many previous cases, people had tried to predict, okay, what are the correlations between the genotype and the total epidemic size? But they had focused in on just one allele at a time. And they had identified, say, that this particular variant, HLA-A24, is a high-risk allele. But what we found that each person has a genotype with 6 variants, not just one. So if we look at these 3 populations, these are the 3 populations with the highest frequency of this high-risk allele. And we found that the average epidemic size for the first ethnicity was indeed pretty high. But for these second and third ranked ethnicities, the epidemic size they experience is very low, 0.009. That's a very small epidemic. So even though 38% of the people have this high-risk allele, overall, the population actually experiences a very small epidemic because they also have other alleles. So maybe there's a compensatory effect where one of those other alleles they have is able to recognize the virus very well. And because of that, their immune response is actually pretty strong and they are not that susceptible. So this was one of the insights that focusing in on just one high-risk allele is actually not enough. We also looked at, is it more important to focus on host genetics or is it more important to focus on pathogen genetics, the virus genetics? So to do that, we looked at the fact that in general, indigenous ethnicities have been seen to experience more severe epidemics than their non-indigenous counterparts. And in our case, the only indigenous ethnicity we had data for was USA Alaska Yupik. And this ethnicity was indeed predicted to experience a worse epidemic than other non-indigenous ethnicities also from the USA. But we also looked at these 2 indigenous ethnicities from Australia, for which we didn't have non-indigenous counterparts to compare against. But we can compare these 2 ethnicities with different viruses. So for these ethnicities, if you focus in on the fact that if you focus in on viruses which are prevalent in the Australia-New Zealand region, we find that the predicted epidemic size can actually be quite small. So for strains isolated in Australia, the trend of which ethnicity experiences a larger epidemic can actually switch from their trend when we look at all viruses in general. So this tells us that not just host heterogeneity, host genetics, but also pathogen genetics is important to consider in figuring out how the final epidemic size will be. So this leads us to a few conclusions. This was actually one of the first tools to incorporate real data about genetic heterogeneity into theoretical epidemic models. The key that we used to do this incorporation was the CD8 plus T cell immune response to incorporate both host and pathogen data. This helped us arrive at more accurate predictions of epidemic size. And we found that both host and pathogen heterogeneity are important in determining the final epidemic size. With the really cool insight that greater diversity leads to smaller final epidemic sizes. So I hope you've been convinced that this was an example where we used medical science along with engineering expertise, specifically in this case, data science, machine learning, and differential equations, to obtain novel insights which are explainable and actionable. I've also been working on some extensions of this project. We've looked at the interaction of SARS-CoV-2 with the immune system. This led to the insight that T-cell epitopes correlate with disease severity. Depending on a genetic makeup, we may be able to predict how severe their experienced disease can be. I've also done some modeling on what happens when there are multiple strains circulating at once, which can actually recombine to produce new strains. What will happen in that scenario? And we found that within host parameters have a greater epidemic impact than between host parameters. So again, not just focusing on one aspect, but looking at all aspects and doing this multi-scale modeling, recognizing that it's a complex system, helps us obtain more accurate predictions. This is actually an ongoing work. We're writing the paper now. Let's change gears a bit and talk about HIV. Some of you may not even have heard of HIV, I can imagine. And HIV has a reservoir in the host, which survives despite the host being given therapy. And this reservoir is actually the main obstacle to eradicating HIV. But let me explain this a bit further. HIV stands for human immunodeficiency virus. This is the virus which causes AIDS or acquired immunodeficiency syndrome. Hopefully you've heard of AIDS. AIDS has entered the human population in the 1980s, to the best of our knowledge, and it has caused over 40 million deaths in that time. 40 million deaths, that's a lot. But it's not an old disease. This is a map of the number of new cases of HIV in 2021 in different parts of the world. And if we focus in on the US, there were over 58,000 new cases of HIV just 3 years ago, 4 years ago. In India, there were over 71,000 new cases of HIV in just 1 year. So HIV is still a very important disease which is affecting people and which we need to understand how to cure this. We don't have a cure for this yet. But to understand how to cure it, we need to understand why it hasn't been cured so far. What's the problem? The way HIV works is HIV is a virus depicted here by this red creature here. It infects a person. The most important cell within the body that HIV infects is a CD4 plus T cell. This is also a T cell. So this may make you think, hey, that's like the immune system. And you're right. This is actually a cell of the immune system. So what happens is HIV enters the body, enters the CD4 plus T cell, and integrates itself into the DNA of the CD4 plus T cell. It then uses the T cell's mechanism, the host mechanisms, to replicate itself. And as disease progresses, the virus keeps replicating, the CD4 plus T cells keep dying, and the person becomes sadder and weaker. But the good news is that we have ART or antiretroviral therapy. Taking usually just one pill a day can actually eliminate all the free-floating virus in the body. And the person feels better, the symptoms are relieved. Unfortunately, whatever virus was integrated into the DNA of the CD4 plus T cell, that remains unaffected. So if at any point the person stops taking ART, this integrated virus can actually start reproducing again. And it can re-establish the infection and the person becomes sick again. So although we have therapy, the therapy does not affect the virus that is integrated into the host cell, and therefore the disease is not cured. So to understand this better, we need to understand this integrated virus. The term for that is provirus a little better. So to understand this integrated provirus better, people have recognized that as the disease progresses, the virus keeps accumulating mutations. So as it keeps accumulating mutations, it diverges. That means its sequence has more and more changes compared to the founder virus, the virus which initiated the infection. So using this, they can now sample integrated intact provirus during treatment and compare it to the founder virus. If they find that the virus is highly diverged, then you can say that, hey, this virus was integrated much later in infection. If the virus is less diverged, it's not integrated late in infection. Now, unfortunately, different groups have found different results. So in this paper, they found that most of the integrated provirus was highly diverged, suggesting that it was integrated near the start of therapy. Some other groups have found a range of divergence values, suggesting that integration took place throughout infection. And some other people have found very low divergence values, suggesting that integration took place early in infection. So you can see there's a contradictory set of results in the literature. And if we don't understand this integrated provirus, it makes it that much harder for us to design therapies which can kill that integrated provirus. So we need to figure out what the answer to this seeming contradiction is. And that's what I did. So we as in people in the literature, had found recently that there are at least two types of CD4 plus T cells, short-lived and long-lived. So that means the immune system itself is heterogeneous. There are different types of cells. Some CD4 plus T cells live for a long time and some live for a short time. So we then wondered, let's use our thought process again, examine the interface between the scales. In this case, the interface between the virus and the host. That interface is this CD4 plus T cell. So if we now factor in the host cell lifespan, can we predict what the divergence of these integrated proviruses will be? Surely the host cell lifespan affects the virus as well. So to do that, we had to collaborate with some experimentalists. This experimental work was carried out in Johns Hopkins here in the US. They worked with 10 macaques and infected them with SIV, which is a variant of HIV which affects macaques. The infection was allowed to develop for 48 weeks, so about a year. So during that time, the virus keeps replicating and accumulating mutations and diverging further and further from the founder. And here we know what the founder is because the experimentalists were the one who delivered the founder virus. Then the monkeys were given treatment and they were maintained on treatment for 4 years. So over 4 years, at different times, different blood samples were drawn. And we then compared the integrated viral sequences, integrated provirus, with the founder virus to see how diverged the integrated proviruses were. And we found something really cool. We found that the divergence is not static, but it changes with time. So when we look at this plot, the x-axis shows us the time on ART, that is the time after starting the treatment. The y-axis shows us the divergence from the founder virus. A larger value indicates high divergence, meaning it's from later in infection. A smaller value indicates low divergence, meaning it's actually very close to the founder, earlier in infection. And we find that there's a biphasic decline in the divergence. There's an initial rapid decline, followed by a second slow decline in the divergence. So I hope you can already start to see maybe the contradictory results are because the divergence is not static. Maybe the samples were drawn at different times and that may affect the results that they get. But that still doesn't quite explain how or why can we then predict what the divergence will be, what is affecting this. So to investigate this, we tried to estimate the mutation profile in short and long-lived host cells separately. Can we distinguish what the divergence will be in the short-lived host cell from the divergence in the long-lived host cell? Remember, the host cell has different subpopulations. So to do that, we have data at the start of ART. We have the distribution of mutations, but this is from a mixture of short and long-lived cells. And at our last time point, which is 3 years later or 4 years later, we have a distribution of mutations. But at this time, since it's 3 to 4 years after therapy, the short-lived host cells would have died out. Only the long-lived host cells remain. We computed that using normal differential equations in math. So we find that at the 3-year time point, since only long-lived cells remain, whatever divergence we see comes from the long-lived host cells. So what we did was we magnified this to account for the host cell death and separated this magnified value from the distribution at the start of ART. So this gives us the distribution of mutations separately in short-lived cells and separately in long-lived cells. So the orange corresponds to short-lived cells and gray corresponds to long-lived cells. So we can see from this plot that for a particular monkey, the monkey is labeled T523. The x-axis shows us divergence, and the y-axis shows us the frequency of that divergence. So this is a normal histogram. If you compare the orange bars, with the gray bars, the gray line here gives you the mean divergence in long-lived cells, and the orange line is the mean divergence in short-lived cells. This is the overall median. We can see that on average, short-lived cells seem to have provirus which are more diverged, and long-lived cells seem to have provirus which are less diverged. So there is a difference in what HIV is present in the short-lived cells and what HIV is present in the long-lived cells. So we wondered, does this difference explain the dynamics of the divergence, that biphasic decline that we saw? So to do that, we use math again. We start with these estimated proportions, these divergences in the short and long-lived cells, and we model how these would change if the host cells were to die at the rate that we can estimate from data. So we allow this distribution to decay with the half-life of the short-lived cells. We allow this distribution to decay with the half-life of the long-lived cells. And at each step, we compute the overall divergence by combining both the distributions of all the surviving cells. And we found that the predictions match the data. In this plot, the red line is the experimental divergence, the same data that we saw earlier. The green lines are the predicted divergence when we simply start with different distributions in the short-lived cells and the long-lived cells and say, okay, given the lifespan, what will the predicted divergence be? And we find that the thick green line, which is the average, shows an overall similar biphasic decline. These thinner lines are when we account for sampling errors. So in the experiment, only 20 sequences could be sampled. But in our model, we have more than 1,000 sequences. So from that 1,000, if we were to sample 20 and compute divergence using only that, what would the divergence dynamics be then? And we see that because of sampling errors, you can actually obtain a variation, different kinks in the predicted divergence curve. So this shows us that factoring in the host cell lifespan can help us understand what the divergence trends will be over time. So this actually gives us a unifying model to explain the contradictory results that were seen in the literature. First, divergence changes over time, and the reason for that changing divergence is the host cell lifespan. So if in the experiment, the sample was taken after the short-lived cells have died out, you will have only cells which have low divergence, giving the prediction that the integration happened early in infection. If the sample is drawn when there is a mix of short and long-lived cells, we will see a range of divergence values, suggesting that integration happened throughout infection. Whereas if the sampling is done while there are a large number of short-lived cells still circulating in the person, the result will be that this is highly diverged, suggesting that the integration happened near the start of therapy. So this helps us explain and understand better what the integrated provirus actually is and how that will change with time on therapy. So if we were to start trying to kill these integrated proviruses soon after starting our therapy or around the same time, we will actually expect to see a large number of highly diverged integrated proviruses. And this gives us directions to start thinking about therapy. So this leads us to a few conclusions. Proviruses, or HIV, are integrated into two types of host cells, having different half-lives. Short-lived cells harbor more virus from later in infection, whereas long-lived cells harbor more virus from earlier in infection. So there's a difference in the integrated provirus. This gives us a unifying model explaining seemingly contradictory observations in literature and gives us guidelines for treatment to try and eliminate these integrated proviruses, which maybe can result in a cure for HIV. So again, we were able to combine expertise from medical science with expertise from engineering. Specifically, in this case, we used bioinformatics and differential equations. I've also used machine learning, but I didn't present that in this particular presentation, which gave us novel insights, which were explainable and actionable. Hopefully, this has convinced you that applying expertise from medical science and engineering together, particularly through multi-scale modeling, can give us really cool insights into health systems in general. This project is also being extended. We are building a detailed agent-based model of within-host HIV evolution. Now that we've understood so many aspects of it, can we build a model for the whole thing? And we have built the model, and this paper has been submitted for publication. Those are some of my ongoing projects, but I'd like to quickly mention a few others. One of my ongoing projects is incorporating human behavior into epidemic models. So remember in the first project, I said we make the assumption that people are well mixed, that any two people are as likely to meet as any other two people. But we know that's not true. People actually have movements which affect who they come into contact with. I'm not likely to meet everybody in my city. I'll only meet a certain subset of people in my city. So we're trying to figure out how this movement affects epidemics. To do that, we are using empirically measured movement data or GPS data. We're also comparing how people move in universities versus in a school versus in a city. Is there a difference? And if so, what are the implications of that for the spread of infectious diseases? I'm also working on ticks and tick-borne diseases. So these are diseases where rather than transmitting from person to person, you need a tick, which is basically an arachnid like a spider, which bites one person and bites another person. And if the tick has that pathogen, then the people get the disease. Similar to dengue with mosquitoes. So it turns out there's a new tick in the US. It's an invasive tick called H. longicornis. And the work I'm doing is to predict the spread of this invasive tick and the implications to potential pathogens. Another project is extending the work on HIV, looking more at antigen-specific effects on HIV proviral dynamics. So antigen basically means proteins from the virus. So if there are other viruses, many people have both HIV and tuberculosis, then how does tuberculosis affect the evolution of HIV? How does HIV affect the evolution of tuberculosis? Things like that. How do different diseases interact to affect the disease progression in a person who has HIV? If any of you is interested in working with me on any of these ongoing projects, feel free to reach out to me. I'll show my email address again towards the end. I'd also like to quickly mention what I'm teaching here at Binghamton University. In the fall, I'm teaching computer programming for engineers, which is an introduction to computer programming using Python language. This course does not need any background in programming. So if any of you come here and you find that you're struggling with programming, you can come sit in on this course and you'll learn everything about programming, starting from how does a computer work, what is a program, how do I open it, to actually doing machine learning by the end of the course. The other course is healthcare data science and analytics. So this is a more advanced course where we teach data analytics tools, including machine learning, but also simple statistics and basic data science with applications in healthcare systems. So we focus specifically on descriptive data analytics, where the task is to identify patterns in the existing data. Predictive analytics, where we use these patterns to predict what will happen next. And prescriptive analytics, which is, okay, if this is the prediction, how can we intervene to help the patients? Remember, this is healthcare. So our main focus is to help the patients, help the public, help people. So we need to have ways to actually help them. And to do that, we need explainable and actionable insights. So that's the focus of this second course. With that, I'd like to thank my collaborators. As you can see, it's a large number of people. This kind of work is very interdisciplinary. We need people who have expertise in medicine, biology, experimental work, and mathematics and computer science and engineering. So it's a lot of work. It's really fun and it results in really cool insights. So if you're interested, please reach out to me. I'd like to also quickly thank funding and all of you for listening. Thank you. Do you have any questions?
Yeah. Thank you, Narmada, for this presentation. Before we open up the floor for question and answer session, I've noticed that Dr. Geeta Manivasan is here. So, Dr. Geeta, we want to open the floor for you, for a quick introduction about yourself and your vision about this seminar series.
Very good morning to one and all, and good evening to all these. The audience here listening to this talk. So, sorry I missed the first part of the talk because I had a meeting with the chairman. It was an urgent meeting. So, I'm thankful to Dr. Mohammed and Dr. Srinath for arranging this first series, first talk of our Binghamton VIT series. I think it was so interesting. Though I came at the later part of the talk, it was I could understand. So it was so simple, and it's not my area of expertise, but I could understand what she was talking and how important the work is, and it's very focused and very interesting. So I thank all of you for this, and I hope the series goes. And now I can identify. I'm sure like many people will look forward to collaborate with Narmada because there are few faculties working in similar direction. But it's a late evening here, 5:30. Otherwise, I think more computer science faculties would have joined this series. We had a long day. Every day it's a long day. From 8 o'clock we start, and so yeah. So what I hope the next talk will be from our side, if I'm right, and this will initiate some collaboration. And I also feel there's a huge potential for Narmada to have a part of teaching. The two courses, not the Python, because Python we also do some basics, but the second data science, I'm sure like it will be and value added to our students here. With this, I leave the platform to you. And yes, thank you.
Thank you, Dr. Geeta. Dr. Kasavani.
Yeah, I just want to thank Dr. Geeta for joining us and for her significant role in creating this partnership with the School of System Science and Industrial Engineering, and I'm definitely looking forward to our continuing collaboration. This is just the beginning, and I think we have a lot of things that we can do together. So it's a great start by Dr. Narmada's talk, and I think there are lots of things that we can do in the areas of research and proposal writing, publications, teaching, and many more. And looking forward to it. Thank you.
Yeah, I think we should thank Professor Srihari for initiating this.
Yes,
with
yes,
that
absolutely.
volume, this would not have happened. So I'm
Absolutely.
not sure if he has joined, but yeah, I think all of faculties are waiting to ask some questions, if I'm right.
Thank you. I think if you have any questions, please unmute yourself and you can ask the question. Or if you have any questions, you can also post it on the chat as well.
Maybe I'll start with the answer.
Yes.
So I'm Dr. Arun Kumar. So I am associate professor at the School of Shrine. So I recently joined, and it was a great talk, Dr. Narmada, and it was very interesting to see a path from being in a different field and learning like mathematical biology and coming into immunological field, and it's really great to see. It's like a inspiration. So yeah. So I would like to ask like you mentioned like short-lived and long-lived cells, right? So actually, the short-lived cells, could it be like a older cells and the long-lived cells could be a younger population of the same cells? Is there a chance for that?
That's a good question. The way we've been thinking about it is that these are also immune cells after all. So a short-lived cell may be one that gets reactivated quickly. So for it to persist for a long time, it has to be like a memory cell. Like when we get vaccines, the memory of that remains for a long time. And because of that memory, the way the memory works is if we get that disease later, the memory cell gets reactivated and then it can fight the disease. So maybe the short-lived cell is the one that's getting reactivated quickly, and the long-lived cell one is the one that's not getting reactivated quickly. But honestly, we don't really have an answer to that yet, which is why the project I'm working on now is to check what are the antigen-specific effects on these cells. But what you suggested is also a very interesting possibility, which we should look into. Did that answer your question?
Yeah, thanks a lot.
Thank you.
Dr. Narmada, and that was an excellent presentation. Thank you so much. You did it so, it was so cool, like I would say.
Thank you.
I just wanted to check with you. You have mentioned about the viral variants, the pro-viral DNA variations that you have seen. And would these mutations that we call, would they be related to resistance? And would the viral load at the beginning of the infection affect your results per se? Because we know if the viral load is high, the chances of mutations are higher, and resistant mutations are higher also. So would viral load, if you had taken the confounding viral load into your equation, what would there be a difference to your conclusions here?
That's an excellent question. So you're right that some of these mutations may be the ones which confer resistance to therapy. So not even in people where the drugs work and there is no resistance, this reservoir can be a source of a new infection. But in some people, this reservoir contains viruses which are already resistant to that drug. So if that happens, or if drug resistance develops, even during therapy, even though the person has not stopped taking the drug, there can be reestablishment of the infection. So you're right, that can happen. And as far as viral load is concerned, That's a good point. So we found that there is variation across these monkeys. Let me see if I have that in an extra slide. So here is the slide where you're able to see now all 10 monkeys, not just one of them. So the monkey that I showed in my slides was this one, T523. But you can see that in some cases, the predictions match well. In some cases, the prediction don't match quite so well. One of the factors that causes this variation could be the viral load at the beginning of the infection. And because we have data from these monkeys, we can now start to factor in the viral load as well. Now, and you can also see that the initial divergence itself is higher. So that goes back to what you were saying. When the viral load is higher, there's many more mutations and there's higher chance of drug resistance. So in this instance, the initial divergence is high, suggesting that the initial viral load was also high. So that's a very good point. We could now start to incorporate viral load dynamics also into divergence dynamics. So that would be more focused on the virus, not just on the host cell lifespan. So that's an interesting direction to go forward.
And in your first study, you have mentioned about diversity, right? Diversity increases resistance or decreases susceptibility. What about diversity of these subjects in this slide? Would that have a effect on your results?
Yeah, so HIV in this particular instance was not transmitted from one monkey to the next, and therefore it wouldn't affect the results. But there is a diversity. So this particular macaque here, T624, you can see that the initial divergence also is very low. In fact, this macaque didn't really establish infection for a long time. The experimentalists had to keep giving it more and more virus for the infection to establish in the first place. So this particular macaque seems to have had some innate resistance, meaning it was less susceptible. So diversity is something that we can see in this experiment as well.
And with this kind of, these kind of studies, you could also, for example, if you could also predict. Now, I've used some amount of data for predicting, infectious diseases earlier on than the other tools, diagnostic tools would.
Oh, fantastic.
So would these tools that you use help, say, for example, a public scientist to know how well a vaccine is uptaken in a population? Would you be able to help public health scientists know that if there is a launch of a new vaccine, or, now we have a lot of resurgence of mumps, measles, rubella, and diphtheria. These are all vaccine preventable diseases.
Yeah.
So can these be used to predict how well the population is taking up, or adhering to vaccine uptake, or whatever the
Yeah, so from the perspective of behavior, this particular project would not be helpful. But from the perspective of biology, we can use the HLA sequence of the person and see whether peptides from the vaccine. So different vaccines have different compositions. Maybe some people's HLA can bind with the composition in vaccine 1 better than vaccine 2. And that is something we can predict using this approach. We can see in this population, most of the people have the ability to bind with peptides from vaccine 1. So it's a better idea to prioritize giving vaccine 1 in this population than vaccine 2. Something like that can be done.
Maybe not with these systems, or these these methods that you have used for this, but do you have any other tools which could predict vaccine uptake? maybe not this. This is very specific to, these specific queries that you have, or the specific research problem. But if my research problem is, can you use, can you use this to find out what or predict, okay, in 2026, this will be the vaccine uptake in this population in South India with previous, say, for example, retrospective data. Maybe not these tools, any other tools, machine learning tools?
Yes, I can see if we had data for what the trends have been over time for people taking a vaccine, we could use time series analysis and forecasting to try and predict what the percentage may be in the future, assuming the trends stay the same. That's not something I have done personally, but I would love to talk to you about it if that's something you are working on. That would be a very interesting and useful thing to study. But what I brought up on this slide is another direction that I've worked on, which is within host, looking at the response to anything, such as a vaccine. How do the expression levels of different proteins change? So we can measure the transcriptome, meaning the expression levels of different genes. That corresponds to the levels of different proteins. And so different proteins interact with each other. In this example toy that you see here, let's say that this protein is very abundant. That's why it's a large circle. This protein is also very abundant. And we know that these two proteins can bind to each other. If both of these are very abundant, the likelihood that they will bind increases just because they are in higher concentration. And because they are now likely to bind, whatever happens as a result of this binding, that result will be more common. So we use this principle, expand it to all the genes in the body, all the proteins and the interactions. So we can compare now, how does the body respond from between a healthy person and a person who has received the vaccine, healthy person who has received the vaccine. What have the changes been? And do these changes suggest that there will be a good response, or do these changes suggest that there will be a poor response? Specifically, what we've used it for is to build a diagnostic biomarker for tuberculosis. So before the person starts producing sputum, there are changes in the blood. So can we use these changes in the blood to predict whether this person has tuberculosis or not? Many patients are not even able to produce sputum, and sputum test is the most common test for tuberculosis these days. So this is not exactly what you were asking for, but you also mentioned early diagnosis, so I thought I'd quickly mention this.
Yeah. Thank you, Dr. Narmada. For everybody who's participating, I have added the contact information for Dr. Narmada in the chat as well. So that if you want to have any offline communications and more collaborations, I think that would be, we highly encourage that. And all these things we are recording this also, and I will also share the link for this Zoom recording with Dr. Geeta and Dr. Yogendra, so that can also be shared with Shine. We will also host that on our website as well. Yeah. Thank you. Any other questions from the participants? Yeah.
I just have a general statement. I mean, thank you very much, Dr. Narmada, for the wonderful talk. Yeah, and your statement that diversity protects population was a wonderful takeaway for me. Have you also looked into the population resistance that comes with the diversity, and you shine some light on how that might be looked into from a perspective?
I don't think I understood your question. Could you elaborate a bit, please?
Resistance, for instance, disease resistant, the genetic variability in a population, and how could that be looked into as well, the variation in the genetics in a population, right?
Yes, so that is one of the ways in which we've done that study. We've looked at the genetic variation from the perspective of HLA class 1. That was the specific immune molecule that we focused on. And if there is a range a diversity in the genetic composition in such a way that there is a wide range in susceptibility, this actually ends up protecting the population as a whole, rather than if there was pretty decent resistance, but there is no spread, there's no variation, that population would experience a larger epidemic than a population which has more diversity. And this particular molecule is actually it's very clean, the inheritance. You get one copy from each parent. So if there is a lot of, marriage in such a way that there is more variation in the two parents, possibly this can result in a more diverse population. And this can also result in spatial locations which have different diversity. If in some places people who are related to each other live together, so then that makes it a pocket in the city where the diversity may be a little less compared to a city where there's no such pockets of people who are related living together. So that's actually an aspect which I have in mind to study. If there are these spatial pockets, can we combine this genetic analysis with these spatial pockets to see how that would affect the spread of a disease? So a lot of cool things can still be asked.
Thank you.
Thank you.
Any other questions?
All right. If there are no other questions, I'd like to thank all of you for joining, listening, and here's my contact again. And Dr. Srinath has also shared it on the chat. Thank you.
Thank you, Narmada.
Yeah.
Thank you. Thank you very much.
Yeah, thank you all for
Thank
attending.
you.
And let's embark on this journey of shared learning and innovation together with this seminar series.
Thank you very much, everyone, for joining us today.
Bye.
Thank you, Dr. Geeta, Dr. Yugendra,
Thank
all
you,
our
Magma.
colleagues
Thank
from
you, everyone.
VIT. Thank you. And thank you, Dr. Narmada, for giving this excellent talk.
Thank you.
Bye.